is Anneke Lucassen. I'm Director for the Centre for Personalised Medicine here in Oxford. This series speaks to key personalities involved in developing the concept of personalised medicine and asking them to reflect on their careers, but also to think about what the next step for the field might be. It's an absolute pleasure to begin the series with a conversation with Professor Sir Walter Bodmer, who is Emeritus Professor of Genetics at the University of Oxford. In your long and illustrious career, you've made major contributions to multiple fields. I can't summarise them all, but I'm just going to give a very broad introduction that you played a key role in the discovery of the human leukocyte antigen system, a set of genes really important in our immune system and increasingly recognised as important in our defences against viruses and cancer. You led pivotal work on population genetics and particular developed cellular experimental work on bowel cancer. And you're credited with launching the idea of public engagement with science and technology. You have been Director General of the Imperial Cancer Research Fund, Chancellor of the University of Salford and Principal of Hertford College, Oxford. Walter, thank you so much for joining us.
It's a pleasure.
Can we start with your very early life? I believe you were born in Germany and came here as a very young boy.
Yes. My father was Jewish, and rather late in 1938, they realised they'd better leave Germany. I was born in Frankfurt, which is where he was a physician. So we came to this country when I was 2.5. So I don't remember anything of Germany, but I certainly remember what happened fairly soon after we came to England. It was clearly quite difficult for my father in his mid-40s to settle down. He was a doctor, and he was very disappointed that I never studied medicine. So that's where I started. And then we settled in Manchester. It was interesting because during the war, they decided they'd better spread the doctors that came in to different places. They wouldn't let him settle in London. And we had connections in Manchester, so that's where we settled. And I went to Manchester Grammar School. I ended up rather by chance in some ways, just doing mathematics and almost nothing but maths and physics at school, no biology. And so I studied maths in Cambridge. And in my second year, we had a lecture course from David Cox, the late and remarkable statistician, Sir David Cox, which was extremely stimulating and stimulated my interest in statistics. And somehow he then made me realise that amongst the courses I could do, I was going to do statistics, would be some given by this R.A. Fisher guy. And I thought that sounded interesting. It was all about genetics. I didn't have a clue what it was. So in the summer before I started that third year, I had thought I'd better read up a little bit, so I read a genetics text. And I read some of his books, which were rather difficult for someone at that time in my situation. And I was just fascinated by genetics. It was something that seemed new and interesting, had an analytical side to it. And within the end of the first term of my third year, I decided that's what I want to do. And I went to Fisher and I said, "Will you take me on as a student?" And he said, "Oh, yes." Well, I already had a first degree in maths, and he'd had relatively few maths students. So, I mean, that's what changed my life completely. So that's how I, in the end, became a geneticist.
You were in Cambridge around the time that Watson and Crick found the structure of DNA.
I came to Clare College, Jim Watson's college, just as he left, right? So because it was April 1953, and I came up in the October of that year. So, but at that time, I didn't know anything about that. It's interesting that my first introduction to DNA was a lecture by Fisher, which was going to be on abstruse mathematical ideas of recombination, but he started with the most clear-cut description of the whole idea of DNA structure and the code. He was of course a close friend of Francis Crick's, because they were both at King's College in Cambridge. Right. Anyway, so that's how I started. The thing that influenced me enormously is that Fisher, in contrast to what a lot of people think, was very interested in the natural world, and he's very keen that if you're going to do statistics, you should know something about the data that you're working on and be involved in collecting it, know how it's collected. So he actually instilled on me the idea that you should be involved in the data collection, the experimental side, if you will, as well as the analytical side. And that's also stayed with me throughout my career. And I strongly recommend nowadays, if anybody's going to do bioinformatics or things like that, that they really need to do the experimental side. It can be collecting their own data, it can be working in a lab, and not just be an analyst of somebody else's data.
That's so interesting, because I suppose that really teaches you that data on its own is potentially quite biased. So you've got to be out there collecting your own data to know what they...
Absolutely. And of course it was Fisher who, amongst other things, taught you how to remove bias. Although it wasn't the only reason he proposed randomization. He also said it gave you a means of estimating errors properly. But you're absolutely right, and that's absolutely key. And again, that stayed with me, even if I'm not quite up to date in modern statistical approaches.
You then went on to work in a Nobel Prize winning laboratory in the US, didn't you?
Yes, I ended up going to Stanford, which is also another major advantage for me, that it worked out that way.
And you ended up working alongside your wife there, didn't you? Tell us a bit.
Yes, that's a different, interesting, related story. My wife, my first wife, Julia, we met when we were at high school. She was at the school opposite, across the road from the Manchester Grammar School. We used to meet at the bus stop looking after our respective kids from the school. And she actually did PPE in Oxford at Lady Margaret Hall, and through that had done a bit of statistics, and actually had a job with a very distinguished economist, Brian Redaway. But the kids got in the way, and so she couldn't quite continue that. So when we went to Stanford, after a relatively short period of time, she got a bit fed up of looking at after three kids at home all the time. And that's the opportunity at that time that came to get involved in the analysis that eventually led to the discovery of the HLA system. And I put it to her that, she could help a bit with the analysis. She knew a bit of statistics. She learned programming. And in fact, she wrote the first simple programming that led to some of our discoveries. So that's how she came to be involved in science with me there.
And then you came back to Oxford in 1970.
Yes,
right?
I didn't come back to Oxford.
No, you came to Oxford.
The only way I'd been in Oxford was because my wife was a student there, and I used to come totally illegally every weekend to visit her at LMH. Both of us, and my wife perhaps more than me, felt that culturally we'd like to get back to the UK if we could, in spite of the fact that people thought we were mad thinking of leaving Stanford. And so when a job came up, which was then to be the first professor of genetics in Oxford, and we thought about it, but somehow I was actually asked if I would apply for the job. So that's when we came to Oxford.
And tell us a bit about, what was the next step after 1970? The Imperial Cancer
Research? Well, in
Research?
1970, we came to Oxford and set up a small department. It was interesting because at that time, the reason they wanted to start genetics is because they had no established chair of genetics. There were geneticists there, and two very notable figures, Cyril Darlington, who was the professor of botany and a notable geneticist, and E.B. Ford, who was an ecological geneticist. So the biochemists and the plant people and the zoologists got together and said, we'd better have a genetics department or something like it, otherwise, where will the subject go? Well, I said, well, I'll do whatever population genetics teaching you want, but I'm going to choose to be with biochemistry because that's where the future is. So that's how it came to be a sub-department of biochemistry. And there we were able to continue with a lot of help from people in Oxford, the HLA work, which by that time my wife Julia had got very involved in and had her own sort of research component of what we were doing. And the somatic cell genetics work.
What was that like working together with your wife?
The best question would have been to have asked her at the time.
Yes.
I mean, we got on very well, and it worked out. And of course, initially I was more of the leader because I started things off, but she became a notable figure in the HLA field in her own right and widely respected. So we managed it. Later, when we were at the Imperial Cancer Research Fund and I was in charge, we had sort of, like what do they call it, meetings with everybody, could ask questions. She would sit in the front row asking awkward questions.
And when did your role in the Imperial Cancer Research Fund start?
Well, that was, I was in Oxford as Professor of Genetics, and out of the blue, I was asked whether I'd be interested in taking over the post as what was then Director of Research at the Imperial Cancer Research Fund. I really didn't know a lot about cancer, but they thought, wisely as it turned out, that maybe genetics had something to do with it.
And around that time you were leading very instrumental work showing that, I think it was bowel cancer tumours showed new antigens.
Well, no, what happened really, we continued, of course, the HLA and the somatic cell genetics work, but I decided that if I was going to be head of a research institute in cancer, I'd better do some cancer research. So by that time, already before going there, I'd anticipated that we started and we're involved in the early days of monoclonal antibodies and made monoclonal antibodies to some of the cancer cell lines. But there was always also at St Mark's Hospital, they were a major centre for colorectal cancer and for the genetics, because that's where essentially the familial adenomatous polyposis, the inherited colorectal cancer, had been not quite discovered, but really turned into a genetic subject there. And I thought, well, that's genetic, so let's start with that and try and see if we can find out where the gene is. So that's really how I got involved in the colorectal cancer side, through the collaboration there. And we started doing family studies, and that's in the early days when you could begin to use DNA markers and enzymatic restriction polymorphisms and so on. And that turned out to be very successful. So that's really what steered me in terms of the cancer field in continuing a major interest in colorectal cancer.
And it's interesting that work now is turning into early immunisations against cancers.
Yes, that's another interesting story. I mean, we got very involved in the early days of making useful monoclonal antibodies that picked up whether a cell was an epithelial cell. Obviously, a lot that were used in the development of the HLA system, partly against the different components of the system. The work with monoclonal antibodies, it was fairly clear. We did early studies with Sir Milstein, who of course shared the Nobel Prize for monoclonal antibodies. One of our first paper on monoclonal antibodies was with him. And it was fairly clear from early on that, following the ideas of Paul Ehrlich at the early beginnings of the 20th century, that might be a way to do something about cancer because of the specificity of a single antibody being able to attack a single point of difference on a cancer cell versus a normal cell. And we did some work while I was at the ICRF at that time already along those lines. But actually the full involvement in a more directed program of immunotherapy came because my first wife, Julia, she died of a breast cancer. She was one of the first breast cancer patients in this country to be treated with Herceptin, which is against the receptor that's widely used still for treatment, which sadly in her case was not effective. But that actually what stimulated me to say, I'm going to take on, by that time, of course, I was retired and working again in Oxford, as a major interest trying to develop immunotherapy with monoclonal antibodies.
What do you think about public understanding of science now, and particularly about genetics? Do you think?
I think that there's been an enormous change in that direction that's very positive. I mean, at that time, there was very little of that. Now you get, they call it public engagement, it's the same thing in a way. And like in Oxford University, I'm on the Sparks, Oxford Sparks thing. There's a lot of things going on in that way. So I think there's a huge improvement in that direction. And of course, science is, you can hardly pick up the newspaper nowadays without something about genetics or DNA and so on.
So think about the public understanding of science. What I sometimes think I observe is that people expect genetics to be very clear-cut, and that if only they know what's in their genetic code, all will become clear. Do you think that's right? What do you think we could do about that?
I think there is a tendency for that, and I think that's because of the way the publicity about what can be done has happened. I think one can teach the basic ideas of genetics, and should, at the school level, and not complicate them with the problems of quantitative genetics and what determines height or IQ, whatever that is. And the basic ideas are not that complicated, and I think it's very important that people understand that. And then to go from that to the idea of, well, what is a genetic tendency? What is the fact that, yes, on the whole, there is clearly a contribution from inheritance to how tall you are, and all sorts of other aspects. And at the extreme end, I always quote musical ability, that it's impossible to imagine that Mozart didn't have something in his genes that made him able to do what he did. I always used to say in a public discussion, well, okay, if he'd been brought up by Bedouins, then he would have never composed the way he did. But I always say if I'd been in his family and been brought up by them, I would never have composed the way he did either. So you can give some idea at that level.
What do you think about government ambitions to sequence everyone when they're born?
I think that I'd start by saying that the most useful thing is when you find very clear-cut, strongly abnormal genetic sequences that you can now do something about. And even if that's only 2 of, say 5% at most of births, that's a contribution in its own right that I think is important. I think it's going to be quite expensive, but I was at a recent genomics meeting organised by the Beijing Genomics Institute where this came up a lot. And, you can now do sequencing for maybe $100 a time or something like that. And my view on that is that the most difficult part will not be the sequencing. It'll be the interpretation of the sequence. Now, a good friend and former colleague of mine who I discussed this with, he said, oh, that's not so difficult. But I don't think that's true. You can pick up the obvious, and even that's not that easy, to pick up the obvious severe abnormalities. Sometimes what looks like a severe genetic change doesn't seem to have the effect you thought it would. Maybe there's another gene that compensates for it. And then when you start getting into the genes, say into the breast cancer situation, and you get genes in the BRCA1 and 2 loci, if they're clearly abnormal, that's fine. But when you get these amino acid differences, it's not so clear, even if they're changes in sequence that could have some effect. Actually judging to whether they do or not is not easy. I mean, if you take another disease, and of course Huntington's disease was a classic example where the genetics is fairly clear cut, and the question is, do you want to know about it or not? If you're going to get it in your 30s and 40s, a very nasty disease. If you knew you could do something about it early, in that case you could do something early to try and prevent it. That may also be true for Alzheimer's. If there are very clear genetic differences there, if there was something that you knew that a person was susceptible, that there may be a certain stage of their life when you could say, yes, we can do something to try and stop that developing. And I'm sure those things will happen, but each will be a very complicated situation to make sure you're doing the right thing, to do very complicated clinical trials, to judge it in different population groups according to the genetic background.
Yes, and finding out about whether you're at risk of those things at birth may not be quite the right timing either.
That's an interesting question. That's the question of what do you do with incidental discoveries. When we first started doing our genetic studies of the British population, before there was a question of doing a lot of sequencing, the issue always came up, supposing you find that they do have a gene that gives you a susceptibility. You are collecting the data on the basis of understanding there wouldn't be any medical implications, no follow-up. But if you find something that's really serious, is it right to tell people in spite of that or not? It's quite a complicated question. So I think the same thing will arise, have to be very carefully thought about when you do the sequencing at birth. You'll have to decide, and it may be up to the parents to decide, and then if the parents decide, how do they know whether that's what the children would have wanted? So I think these things have to be thought about quite carefully. And so I think in any case, are you going to ask people whether they want to have their children sequenced at birth or not? That's the first question, I presume. The second would be, what about consent? If we do find something, do you want to know or not? So I don't know how much, I'm not involved, but I'm sure that you must be thinking about that a lot, because these are the really key questions. Much more than the technology of sequencing, which now fantastically is so straightforward.
I think that's so interesting because the technology is straightforward, it's the interpretation that's complex.
Absolutely.
But you highlighted earlier there's one set of genetic problems that are really quite strong risk, and then another set of problems that are lots of weak factors throughout the genetic code. And that work really came out of your early work, didn't it? The HLA work. Can you tell us a bit about how that happened?
Well, I should say a bit about HLA, which is said to be human leukocyte antigen. It's interesting that because the A originally stood for the first locus, but that's the way it come to be now. So this arises out of the fact that let's start with a blood transfusion. You can't, if you do a blood transfusion, everybody knows their ABO blood type. And if you go against that, you've got antibodies that give you a nasty reaction. If you do that, and that's because of a genetic difference between people. Now, if you extend that and say, what about if you do a graft? And people know nowadays about bone marrow grafting, about kidney grafts and all of that, and it's widely known that you just cannot take one person's kidney and give it to another without doing something about the fact that they'll be rejected because there are genetic differences that lead to that. So that led to a search for how could you find the genetic differences between people that are not just on the red cells. They've got to be on the surface of virtually all sorts of cells in the body. And that started with people taking blood from someone who'd had many blood transfusions on the assumption that might lead to making antibodies against other people's blood types, and you could use those for seeing whether they gave you a pattern of that recognized things that you could study as genetics between people. They discovered that when they looked at serum from women who'd had multiple offspring, they found antibodies that reacted with the offspring and with the father, but not with their own. And they realized that what that was happening is that was giving rise to antibodies that were limited to the difference between husband and wife, which made it much easier to sort out what was going on. Because if you've got the input from lots of different people and it's variable, you can't sort it out. It was then very easy to work out that if a set of sera tended to react together, they were detecting something in common, and you could find out whether that was a particular antigen and inherited. And that's what led to the discovery of the HLA system. So that led to a huge splurge of studies on associations between HLA and types and disease. What does that mean? You say, we've got a disease, it's rheumatoid arthritis or diabetes. Is there a particular type that's much more frequent in people with the disease than in the control? And if there is, then that suggests that, or as we pointed out in terms of the interpretation, maybe something at a very closely linked low test could be the. And that's really the whole basis for GWAS. Genome-wide association studies are just built on that idea, generalizing it, as we suggested, naturally one could, to doing that not just for HLA, but for any difference. And that's, of course, been a huge development. And some of the interesting discoveries early were very, very significant. I think what's happened subsequently is people are more and more keen to find something. The larger the population you look at, the smaller the difference you can find. You get to a level, yeah, maybe that's something that matters, but we can't really find out because the effect is too small. So that's a long answer to a simple question.
But so interesting, because those GWAS, genome-wide association studies, have really flourished and expanded over the last 20 years, haven't they? And now there's a large study in the UK of Future Health that's recruiting 5 million people to do genome-wide association studies in them. What do you think of that?
I don't think they're going to lead to very much. I think the trouble is that what they call the low-hanging fruit, the things that are interesting, are picked up very easily and quickly. And if you've got even a 20% increase in risk, there's a chance that if you've got a particular genetic variant, it does that. You might find out about how it works and so on. But if the increased risk is only a few percent, even if it's significant, the chances of your finding out why that does something are not very good. And, you know, if you happen to find out that things that affect some sort of behavioral difference to do with brain function all happen to be in genes that are functional in the brain, that doesn't tell you anything new, really. I think it's very difficult. So, of course, what people have got to is this notion of polygenic risk scores, this notion that, okay, if you have only one or two, you can't do anything with it. But if you look at a whole collection of these, and if individuals tend to have a higher proportion of those that have some little effect, which collectively may be a significant effect, is there something you can do with that? So if you find that this risk score makes you more liable to have heart disease, then maybe is there something you can do about it? And in some situations, that's very clearly possible. In the cancer field, everything, if you can detect cancer earlier, it's always an advantage, because if you can detect it earlier in some way, you can usually do something about finding whether it's really there. And an early cancer is always much more curable than a late cancer. But even that's proved to be very difficult, because there are a lot of other factors that might determine whether you get an early colorectal cancer. And it's not clear that these polygenic risk scores add that much to it. And furthermore, they're dependent on the distribution of the sequences of these different variants, which varies from one population to another.
What are you most optimistic about for the future of personalised medicine?
I think the more we understand about even the things that are extremely defective might lead us to be able to understand things that are less so. So if you find a gene that when it's thoroughly knocked out causes a severe abnormality, then something I suggested some years ago, we called it rare variants. There may be other versions of that gene that have lesser effects, but still teach you about something you should do with it. And so I think we should learn more and more from that. And in the end, genetics in some respect lies at the bottom of almost everything you do in biomedical science. But in ways that people don't, it's not the same thing to manipulate an antibody and find a target that's on a cancer. It actually depends on a lot of genetics, but it's not the way that people think about the genetics. So I think that the whole development of genetics just underlies there's nothing we could do about improving treatment. Of course, if you want to do, even with prevention, you want to understand that there are a lot of things where it's the social setting that matters. And so I think so long as precision medicine isn't interpreted too narrowly, and so long as it's really used as a means of supporting genetic studies, and the information you can get, the more you can get, the more you're going to be able to help, that's fine. But I think it's important that it not be given too much of we're just going to sequence everybody and we'll solve all their problems. It's not going to be like that.
Walter, an absolutely fascinating conversation. Thank you so much.
My pleasure.